Okay, so here we're going to talk about enzymes and a little bit more on their regulation. So when you talk about regulation, things that you have to consider are not only whether or not the reaction needs to go forward, but also what is controlling the reaction. And oftentimes things distant from the reaction are controlling it because those distant things are downstream. And when distant things downstream accumulate, they want to stop the steps before them to keep the reaction going forward. So we have in general two categories. We have reversible and irreversible inhibitors. So your reversible inhibitors are going to be of two types. We're going to have our competitive and non-competitive. And they are what they say. They're reversible. One will compete with the substrate and one will not compete with the substrate. Now the other category of irreversible are usually going to be things like suicide inhibitors, in which you have a covalent modification to the enzyme and it cannot be reversed. So the enzyme cannot function. So basically you lose the functionality of the enzyme. An example is cyanide poisoning. And cyanide is going to be bound, is going to bind to ferric. And the enzymes that are going to use ferric will become inactivated. This is the ferric form. So that's just an example. Now we also have regulation at the level of conformation changes. And so there's a few types of this. One is allosteric regulation. And another is when you have covalent modification. And covalent modification we've already talked about. This is an example of phosphorylation. And of course, these are all reversible. Allosteric is simply going to mean that you have an alteration of the protein by a different effector molecule binding. Protein. And these effectors are, of course, going to be molecules themselves, and they can either be activating or inactivating. And then as far as the amount of enzyme, regulation of the amount of enzyme, this can be in the form of synthesis, making more of the enzyme protein, or in degrading, increasing the breakdown of the protein, decreasing the breakdown of the protein, increasing the synthesis of the protein, the enzyme, or decreasing it. Now, reversible, competitive, and non-competitive inhibition are a little bit different. So competitive, which is shown on A, is going to bind to the actual catalytic site. And this is going to compete with the substrate. So if you look at a Line-Weavenberg plot, this is going to have no change on Vmax, so this is unchanged. However, the Km, or the amount of substrate needed to reach 1/2 Vmax, is going to increase. Now in a non-competitive, this is where the substrate, or the competing, the competitor binds to the enzyme substrate complex or to the enzyme itself, but it can bind to an enzyme substrate complex at a site different than the substrate that's bound. And so this is an example shown here where you have A and B, and A will bind to your substrate, and this Ni will bind to this portion that's supposed to bind B. And so it's a non-competitive inhibitor of A, meaning that the products are not going to be made because it no longer allows B to bind, for example. And this is where it cannot be reversed by the substrate. So that means no matter how much of A you add, it's not going to alter it. So in this case, your Km is unaltered, and your Vmax decreases. And the decrease is going to be dependent on the amount of the inhibitor. Now here we have a competitive inhibition on the Lang-Weavenberg plot. And some things to note, of course, is that the Vmax has not changed. So you can see with the inhibitor and without the inhibitor, Vmax is unaltered. And here you can see that the Km has changed. The Km has increased. Now remember, we're looking at this 1 over Km. And so one thing to keep in mind with competitive inhibition is that most of our drugs are going to be altering this type of enzyme. So most drugs are competitive inhibitors. And the reason is because you have 2 routes to reverse the drug. And the first route, of course, is to decrease the inhibitor. And decreasing the inhibitor will allow more substrate to bind to more free enzymes. And the second, of course, is you can increase the substrate. And this will be outcompeting the inhibitor for more bound enzyme. And both of these 2 routes to reverse it will increase the amount of product that is made. Now in a non-competitive inhibitor, you can see here, in this case, the Km is unaltered. That's at x-intercept. And the Vmax decreases. And so this is where you have an inhibitor that binds to a site distant from the substrates, and the competitor can bind either the enzyme substrate complex or the enzyme. So you can have an enzyme bound to the inhibitor, or the inhibitor is going to bind to the complex of the enzyme and substrate. Okay, so now some definitions here. We have allosteric. Allosteric is in which your enzyme is going to have the site in which a inhibitor or an activator can bind to. And so many of the enzymes we're going to talk about are going to be activated by certain things like ATP or inhibited by certain things like AMP. And all that really means is that that enzyme can bind ATP, and that ATP will open up the enzyme site so you can make more product. Or that same enzyme might bind ADP or AMP, and it closes so you make less product. So it's going to induce a conformational change in the actual enzyme. Now there are also isozymes. Those are the ones that can have different enzymes in different tissues, and they all have the same substrate, same product, and they can all have different KMs, so different affinities. Remember, KM is affinity. And so we'll see that with respect to our glucose receptors. We can also have proteolytic activation of our enzymes. So our zymogens, which is really a proenzyme, are going to be irreversibly converted to an active form. And examples of zymogens are trypsin, chymotrypsin, that get released inactive and they have to get activated. There's also the covalent modifications that we've talked about, such as adding phosphate groups And note that this is reversible. It's not one of those suicide type inhibitors that we just discussed. So let's just talk about a little bit about allosteric activation. So the first is a sequential allosteric activation. And so just keep in mind as we talk about enzymes, it's very rare for the enzymes to proceed in isolation. They're going to catalyze individual steps in a multi-step pathway. And many times these pathways have multiple branches. And so any given enzyme is going to be dependent on activating a precursor reaction steps. So again, if you want to stop a process from going forward, you're always going to go back to that first point where you can control the process. And all the steps in between will be subsequently also inhibited or activated. So for a sequential allosteric activation, this is going to be an enzyme that is multimeric. And this simply just means that it has 2 or more subunits. We talked about the hemoglobin, and that was the example, and it still is an example. And then so the first substrate binding is going to be hard. But this has a conformation change from taut to relaxed. And then your other substrates bind easier. And so in your taut form, the enzyme is inactive because there's no substrate. And in the relaxed form, the enzyme is active and substrate is bound. Now, allosteric activators or inhibitors are going to, again, bind to a site outside the active site, and they are all going to cause conformational changes. Now, the activators, let me do white. Activators bind tightly to the relaxed state. And inhibitors are going to bind to the taut state, or the non-relaxed. And so when you do have an inhibitor bound, you're going to have to either increase the substrate or increase an activator to overcome the inhibitor. And so this is just a model of allosteric enzymes. So here we have our taut state and we have our inhibitor bound. And here we have our relaxed state, which has our activator bound. And then our activator is going to enable your substrates to bind. And so basically, without any activator or inhibitor, you have an in-between curve. Whereas if you had an inhibitor, you're going to need to increase the concentration of your substrate. And then if you have an activator, your activator is going to allow more substrate to bind faster, so it's going to have more of a hyperbolic curve. Okay, so now with respect to covalent modification, you're going to have, for example, phosphorylation that can activate or inhibit enzymes. So our example here is phosphorylation. And this can again be activating or inhibiting. And often it is doing this in opposing pathways. Okay, and so we have our protein kinases, and protein kinases are going to add phosphates they're going to take a phosphate from ATP, and they're going to add to the OH group of your serine, threonine, or tyrosine. So all 3 of these have an OH group in which you can add a phosphate to. And then our protein phosphatase is going to remove, it's going to cause conformational change, and it's going to remove that bulky phosphate group. Okay, and so again, your kinase is going to phosphorylate, your phosphatase is going to dephosphorylate, and they can impact different enzymes. Your phosphatase, by removing a phosphate group, will turn on some enzymes, turn off others, and your kinase will do the same thing, turn on and turn off. They usually are going to turn on and off opposing enzymes. Now, muscle glycogen phosphorylase is going to be important in glycogen degradation in muscles. And so it's going to facilitate a rate-limiting step in glycogen to glucose 1-phosphate. And so it's going to get activated in several ways because your muscle has to tap into energy as needed. So there's going to be an allosteric activator of AMP. And AMP, you can see, is binding to a site that's distant from the substrate. So these S's mean substrate. And so your AMP is going to bind, and it's going to open up the substrate binding site. So here's the substrate closed, and then your AMP is an allosteric activator, so it binds distant, but it causes a conformation change. And so now that substrate site is open, and it can bind to glycogen, and it can start degrading glycogen. And then you have here, you have activation of phosphorylase kinase. And if you have activation of your phosphorylase kinase, it's going to add a phosphate group, again, allosteric, at a site distant than your substrate, but it has the same effect. It's still going to open up that substrate site, so substrate can bind, and you can start breaking things down. So regardless if it's AMP or a phosphate, both of them are going to have the same effect on this enzyme. And you can see that through the drawing, they're trying to show that they're binding to different sites because AMP looks very different than your phosphorylation. And remember, your phosphorylation is going to be on those serine, threonine, and tyrosine groups, whereas your AMP has to bind to its actual nice little pocket. Now, another example is protein kinase A. Protein kinases sometimes are going to be dedicated, and they're going to be regulated by only one protein. So pKa is a serine, threonine protein kinase. And it's going to have a coordinated response by phosphorylating many enzymes. In one of the enzymes it does phosphorylate is glycogen phosphorylase kinase. So what's going on with your pKa sorry, is that you're going to have a signal. So your signal can be adrenaline, which is also epinephrine. Adrenaline is going to increase cyclic AMP, which is shown here. Here's your cyclic AMP. And then your cyclic AMP is going to bind to your regulatory subunits of your protein kinase A. So here's your protein kinase A inactive. You have your catalytic units here, and you have your regulatory units here. And when you have activation of cyclic AMP, or it's not activation, it's actually you make it. So it's very different. It makes cyclic AMP. Your cyclic AMP can then bind to the regulatory site, and it dissociates from the catalytic site. And then your catalytic site is able to go and phosphorylate different enzymes, such as glycogen phosphorylase kinase. Now, we've already talked about other covalent modifications, and these are going to affect things like the activity of the enzyme or where it's going to go in the enzyme. So lipids can be added, as we talked about before. You can have regulation of phosphorylation, acetylation, adding nitrogen groups, adding ribose, ribosylation, etc. A lipid is going to be added if you're going to embed a protein in the cell membrane, for example. And so that's going to help cluster areas around signaling receptors so that the downstream signaling pathways are very localized. Now, some other things that are going to happen are you're going to sometimes have protein-protein interactions that are going to cause conformational changes. So calcium calmodulin is a protein-protein. Interaction, and you're going to get conformation change at the active site, which is also referred to as steric hindrance. And so you're going to have, with the case for calcium calmodulin, you have your modulator protein and calcium that bind in the cytosol. And the hinge region is going to fold over during the regulation. So when you get calcium binding, your hinge region will fold. And so what's causing all this is you're going to have a nerve impulse. And this is going to lead to calcium release from your sarcoplasmic reticulum, for example. And you release calcium. And the calmodulin subunit of muscle glycogen phosphorylase kinase is going to bind to the calcium. And you're going to get the hinge is then going to change conformation, and you're going to get your phosphorylation of glycogen phosphorylase, and the muscle will contract. Now, calcium is also going to have the added effect of binding to troponin C, which is a regulator of the troponin complex, which is also important in the contraction of your muscles. So calcium in muscle has dual functions of interacting with the calmodulin and affecting its conformation and also in binding to troponin C. So I'll put it here. Now, another type of control is on G proteins. So G proteins are small, they're monomeric, and they're what we call reversible masters of regulation. Now, they are going to affect the conformation of other proteins. So, and a G protein binds GTP, and it's called a G protein because it interacts with GTP. So here it's showing it binding GTP, And this is going to be active, and it's going to bind to a target. Now, binding to this target can activate or inactivate. So the active GP binds to its target, and then it has sort of an auto catalytic activity in which you have a bound GTP, and then it's going to lose a phosphate, and you're going to have a GDP, and this becomes inactive. So if you lose a phosphate, you get inactive G protein. Now there are some things that can regulate G proteins. So we have, for example, a protein called GAP, which is going to increase the rate of GTP hydrolysis. And so it increases the rate of G protein target dissociation. And then you have another protein called GET, and this is going to bind to the G protein and increase the rate of GTP exchange. And so it's going to activate the G protein. Now, RAS is a very important small G protein, and it has within it its superfamily. And so the superfamily has a bunch of funny names: RAS-Rho, RAF-RAN. And so what's really important about the RAS network is that they are important in growth and morphogenesis, motility, trafficking, nucleus, etc. And so all of these fancy things are very important in cancer development. So RAS is actually a proto-oncogene. And we just talked about your G protein, it's a little blob, and with a bound GTP is active. So one way for this to be a proto-oncogene is that you cannot convert this to a GDP. So it stays on. And so if it's modulating growth, for example, your cells are just going to continue to grow. And so G proteins, especially the RAS family, are important for cancer. Now there's also regulation in the amount of enzymes, and this can be again in the form of synthesis. So again, we know that in the nucleus, we encode all of our enzymes. So if you have a need for increasing the enzyme, it's going to go back to the DNA, and you're going to translate more of your enzyme protein. Some other ways you can do is so we'll say increase activity. So you can do it through an increase of synthesis, or stability. Your messenger RNA can be stable, so it's not degraded very quickly. And all of this activity to increase synthesis, this is of course very slow. So this is going to take hours to days. So it's not fast like that phosphorylation, dephosphorylation on-off. Now for a decrease, you can increase your degradation. And so proteins are usually going to be degraded, and they usually have a set half-life, and they can be degraded in a lysosome, which digests proteins, or they can be targeted to a proteasome, which of course degrades proteins. An example is during fasting, protein degradation in your muscle tissue is going to be activated, and this is in order to increase the amino acids necessary for gluconeogenesis. So in fasting, your muscles will degrade proteins, and they'll be released to liver for gluconeogenesis. So for our proteasome, these are going to be ubiquitin-tagged proteins. So our proteasome is sort of a barrel, and then you have some protein. Here's your protein. And then your protein is going to be tagged with these little yellow dots, and this is ubiquitin. And then this will feed it, and then you'll get on the other end peptides. So your proteins will be degraded by your proteasomes and become peptides. Now, with respect to regulation pathways, there's a few concepts that we have. We have metabolic pathways, of course, are going to be sequential. And so these reactions, you have an enzyme making one product, but it usually is dependent on the reaction of the previous step. And so they're going to, regulation of these pathways are usually going to occur in what we call rate limiting steps. And these rate limiting steps are usually the point at which there's no return, or it's a branching point. You need one thing over another thing. And so those steps are going to be key in making sure that your cells are making the right products that they need. So we have feed-forward regulation. And feed-forward regulation is going to be important for toxic. And this means if the substrate is present, the reaction proceeds. So if alcohol is present, your enzymes are going to metabolize it. They have to. It's a feed forward. Now we also have feedback inhibition. And our feedback inhibition is shown here, where you have product inhibition, in which product B will inhibit the making of more product B. You can also have it distant. So this is a branching point here. So product E can feed back and inhibit enzyme 2, so you push everything through enzyme 5. Or you can go back and the product can go back to the nucleus and actually inhibit more production of a particular enzyme. So that's a feedback. And this is going to usually involve allosteric regulation of the rate-limiting enzymes. Now we also have, we're going to talk a little bit more about tissue-specific zymogens, so, or isozymes. So we have tissue-specific isozymes. And we have counter-regulation. Now, the tissue-specific isozymes we talked about, we have different forms of the same enzyme in different tissues that have different KMs. An example is glucokinase and hexokinase. And our counter-regulatory pathways are where you have synthesis and degradation of a compound may differ by one step. An example for this is going to be glycogen synthesis. and glycogen degradation. Now, another thing to keep in mind is that these pathways have to be able to find each other. So you have compartmentalization, in which your enzymes are going to be found in a certain area. So a good example is all your TCA cycle enzymes are going to be found in the inner membrane of your mitochondria, for example. Now, we also have enzymes that are going to be important for diseases. So in these particular enzymes, you're looking for something in the serum that's going to indicate some sort of damage. And as I mentioned before, these enzymes are released because your actual cells are dying and releasing them. Otherwise, they would be kept in the organ. And so you can measure these components into the blood. So, for example, when you're measuring liver cell enzymes, you're looking for liver cell damage. When you're measuring heart, you're looking for heart damage. So for liver damage, we're going to look at ALT and AST. And these can be diagnostic because the liver expresses a lot of these enzymes. And so you're going to have alanine transaminase. And so you're also going to have aspartate transaminase. And so you're going to need aspartate transaminase is going to be found in your cytoplasm. So it's a cytoplasmic enzyme. And it also has a mitochondria version, and it's going to participate in what's called the malate aspartate shuttle in the mitochondria. And then alanine transaminase is primarily going to be found in hepatocytes, and it's associated with urea cycle. So our ratio of ALT to AST can be diagnostic. And so if you have a liver disease with no viral origin, your ALT, AST is less than 1. Okay, so that means you have more AST than you have ALT. If you have liver disease with a hepatitis virus, so you have a viral hepatitis, and then your ALT over AST will be greater than 1. So now you have more ALT. And so ALT, remember, is going to be primary in the liver, so your hepatitis virus is killing liver cells and releasing ALT. Whereas no viral origin, you have still your cells in your body releasing AST, and you're not having that massive death that's releasing the ALT. AST can also be a measurement for heart. So following a heart injury, your AST levels rise within 8 hours. So if I do, oops, a little graph here. So you have 8 hours, and then you have your AST. So your AST levels will rise within 8 hours, and then it'll peak at about 24 hours. So let me make that not quite so sharp. I'm running out of room. And then it'll return to normal within about 7 days. So this will be 7 days. So AST cannot be diagnostic for myocardial infarction, but with a combination of others such as your LDH, lactic dehydrogenase, as well as creatine kinase measurements, it can be added to that combination. So another enzyme that's important in diagnosis is cardiac troponins. These are a complex of 3 regulatory proteins. You have troponin C, which is going to bind to that calcium. You have troponin I, and this TNI in skeleton is fast twitch. And then you can have slow twitch. And then you can have a version of cardiac. And then you also have troponin T. Now, troponins are going to be attached to tropomyosin, and they're going to be found in the grooves between thin actin filaments as well as with your striated muscle tissues. It's going to be found in only skeletal and cardiac muscle. So cardiac troponin, troponin I and T are markers for myocardial infarction and damage to your heart muscle. Now, lactate dehydrogenase we've seen before, and so remember there are going to be 5 different isoforms. The M form is going to be muscle specific. So we have our M form. And then we have our H form that's going to be heart specific. The M form is going to be encoded by the LDA gene. And the H form is by the LDHB gene. Humans also are going to be able to express LDHC, but this is restricted to the testes. And LDHD is a mitochondria enzyme for D-lactate. So that came from bacteria. And then, of course, we have creatine kinase. So creatine kinase is also called creatine phosphokinase, CPK. This is going to be found in heart and skeletal muscle. And the brain. And so you're going to have CPK3, which is also CPKMM. It's a homodimer. MM is a homodimer, same thing. And it's going to be found mostly in muscle. And it's going to be found in normal serum. And then you have CPK2. which is CPKMB. So this is a heterodimer. And this is going to account for 35% of cardiac CK activity. And then you have CPK1, which is going to be CPKBB. Then this is going to be the brain isoform. And this should be 0% in normal serum. So in normal serum, you're going to find 100% of all CPK proteins should be just your muscles. You should not find heart and you should not find the brain versions. So after a myocardial infarction, you're going to have the CPK-MB released. And so this can be helpful in diagnosis. Now another one is gamma-glutamyl transferases. And this is a family of enzymes that's going to transfer glutamyl groups of glutathione to amino acids. These are very important for moving things through the blood. And so there are going to be 5 primary GGT genes. You have GGT1, GGT2, GGT5, GGT6, and 17. The genes are post-transcriptionally cleaved, and they have heavy and light chains, and the heavy chain anchors the protein to membranes, and the light chain has the actual catalytic function. So elevated serum GGT levels occur during disease of liver, your bile system, and your pancreas. And then low levels circulating are associated with cardiovascular disease. So an increase levels indicate your liver, biliary, or pancreas. And all 3 of those are kind of linked together. A decrease is going to indicate cardiovascular. And then finally, we have carbonic anhydrase, which we've talked about. So this is going to be that guy who converts carbon dioxide to carbonic acid. And so there are 5 classes. We have our alpha, we have our beta, gamma, delta, and an epsilon. And in mammals, class alpha is expressed. Humans have 15 isoforms. We have the 2 that are expressed in our erythrocytes are calcium anhydrase 2 and calcium anhydrase 1. Now 2 is also found, so both of these are found in RBCs, and 2 is also going to be found in cerebrospinal fluid. And so it can indicate brain disease. And we have carbonic anhydrase 3, and this is going to be found in your heart. So again, it's associated with myocardial infarctions. You have calcium anhydrase 9, and this is going to be in the gut, and so it can be associated with renal cell carcinomas. And normal renal cells do not express carbonic anhydrase 9, but one that is cancerous does.